hybrid cross was done, did I explain about the... Yeah. All right. So today we're going to see the next few things, some different crosses that we have to discuss. The first one is a test cross. For example, you have been given a certain organism, okay? And you don't know the genotype of that organism. So now in order to find out the genotype of that organism, what you will do? You will do a test cross. And what do we do in a test cross? Basically, we take the dominant phenotypic organism and then we are going to cross it with the recessive pairing. Okay. In order, like we know that to find out the F2 generation, we do self-pollination, right? Selfing. But here, instead of doing that, we will cross the dominant phenotype organism with the recessive, homozygously recessive pairing. And this is what we call as the test cross. And this test cross is done so as to find out the genotype of the organism. Understood?
The test cross is used to find the genotype of the organism.
Yeah, to determine the genotype of the organism.
Okay.
Now, after that, up until now, we have seen a lot of Mendel's law and we have seen how the whole entire inheritance takes place, right?
I'm really sorry. Could you say how the test cross is done again? Like how it is done?
For example, we are saying like in order to determine the genotype of the organism, unknown genotype. So for example, instead of finding out the F2 generation, which like if we take the example of the height, we used to do this, right? We used to take two members of the F1 and cross them together. But instead of doing this, what you're going to do? You're going to do this with the recessive parent.
Okay.
And then you're going to find out the F2. Yeah, this is basically what, and they'll not ask you to do so, but this is like a theoretical thing that you have to understand. Is it clear?
Yes.
Now comes the exceptions of the Mendelian principles or the laws. Now, up until now, we have seen two laws given by Mendel. We have seen how the process of inheritance takes place, right? Now, moving forward, we are going to see two such examples that violate the law, that do not follow the Mendelian's law. The first case is incomplete dominance. Like the name only suggests, dominance is there, but incompletely it is there. Now, this one is seen in the flower color of the flower that is called as snapdragon, or many a times it is called as antherhinum or dog flower or 4 o'clock flower. All of these are the names. Now, what happens over here is that the dominant flower color is red and the recessive flower color is white. But when they are crossed together in the F1 generation, like normally, what does the Mendelian law says? It says that in the F1 generation, the offspring is going to be dominant, right? And it says that in the F2 generation, both dominant and recessive traits are going to be seen. But here, instead of the dominant trait being shown in the F1 generation, there is an intermediate trait shown that is red, white. No, instead of that, pink colored flowers were seen. So this is like completely very different from what we have studied up until now. Again, if you do the, like if you find out the F2 generation, so at the end, you will find out that there is one red flower, two white flowers, like the heterozygotes. Instead of showing the dominant character, they are showing the intermediate character. And then there is the one recessive character. So all in all, the phenotypic ratio and the genotypic ratio comes out to be 1:1, which is like not what we used to get in the normal monohybrid cross. Understood?
Yes.
So this is one such exception of the Mendelian's law. Second exception is the case of codominance. Codominance, you understand like how co-education is there, male and female both studying together, that is co-education. Same way, codominance means that two traits will be expressing themselves together. Instead of one being dominant, one being recessive, or one expressing and one getting masked, no, both of them are getting expressed together. For example, can you tell me how many blood groups are there total?
4.
4. A, B, and O, right? Now see, A blood group is single, B blood group is single, and O blood group is single. But AB here, A is also being expressed and B is also being expressed. So that is the case of codominance. Okay, when both the alleles exist and express themselves equally. The best example for this is the ABO blood group of human beings. What is your blood group?
B positive.
B positive. Okay. Now see, the blood group that is determined in every organism, that is dependent upon the antigen the RBC is going to carry. Now see, you know our RBCs, these are the red blood cells, right, in our body, and they carry the hemoglobin and everything. Now, on the surface of the RBC, there are certain proteins present, which we call as antigens. Okay.
On the surface of the RBC.
Yes, on the surface of the RBC, antigens are present, which makes antibodies. Okay. Antibodies are those proteins that fight the infectious diseases in our body. Okay, not just diseases, any foreign particle that comes inside our body, the antibodies fight them. Okay. Now,
all the RBCs contain antibodies, no, antigens.
not
and
all.
these, not all.
Not all. Not all of them. Like, I'll show you how. See, for example, this is the whole entire chart of how the blood group gets expressed. But here, see, every particular character has some sort of a genotype. So, for example, if I'm talking about the blood group A, so IA, this is a homozygous condition. This is when the blood group is going to be A. IB, this is when the blood group is going to be B. Okay. Then IB, this is when the blood group is going to be AB. And when there is no antigen at all, small I, this is going to be the blood group O. But if you have this condition also present, like this, then the A is going to be dominant and the A will express it. Understood? And when this condition is going to be with B, like that, then also B will express. This is clear?
Yes.
Okay. Now see, if the antigen present on the RBC surface is A, okay, the blood group is A for that person, and the antibody it will make, it will be against the B blood group. So for example, if the person with blood group A gets the blood from the person with blood group B, then the A's blood group is going to attack the B. And then clumping will happen in the body. Okay. So the surface of the antigen when it carries the surface of the RBC when it carries the antigen A, then the blood group is A, and the antibody it will make, it will be against the B. Clear? Okay. If the surface has the antigen B, then the blood group is B, and the antibody it will make will be against A. Okay. These two cases. Okay. Now, if the surface of the RBCs have the antigen AB, then the blood group will be AB. But now here comes the catch. They do not synthesize any antibody. Why? Because imagine it like this. If they are going to synthesize A antibody, then also it will be attacking itself because it has A in it also. If it makes B antibody, then also it will be attacking itself because it has B in the blood also. So that is why the people with AB blood group, they do not have any antibodies made against any blood group. Okay. Lastly, there comes the O blood group. So they do not have any antigen present on the surface of RBC. No antigen. But they make antibodies for A and they make antibodies for B as well. Again. Okay, so now, all in all, if we were to conclude this, the person with the blood group AB, since this person doesn't make any antibodies, so it can take blood from anyone because there's nothing to attack any kind of blood type, isn't it? Okay. So that is why we say that the person with the blood group AB are said to be universal recipients because they can take blood from anyone. They can take blood from O, A, B, and A, all 4 blood types.
So, any diseases or any infection comes, how will the body like survive? Because there's no antibodies, right? So how it will fight?
This is not for all the different types of pathogens. This is particularly for the blood type. This antibody is for blood type. There are like thousands and thousands of antibodies in our body specific to everything. But here, These ones are specific to the blood type. Clear?
Okay.
Hmm.
Yes, sir.
So since they do not have any antibody against any of the blood groups, so they can take blood from anyone, and that is why we call them as universal recipients. Whereas since the O blood group doesn't have any antigen present, it means that it will not trigger any reaction in anyone's body. So that is why we call the O blood group as the universal donor. Understood?
Okay.
This blood group thingy is clear?
Yes.
Always remember that the person with A blood group can take blood from A and O. B blood group can take blood from B and O. The O blood group can donate to any of the blood group, but it can only take from O.
Okay.
Clear?
Yes.
So this is like variety of the process that you can practice that I've written over here. Okay. Now, moving forward, talking about the inheritance of two genes, which we call as the dihybrid cross. Now, when we talk about the monohybrid cross, we were only taking one character into consideration, right? We just took height, or we could have taken seed shape, or we could have taken seed color. But now we're going to take two characters together. For example, if we're going to take seed color and seed shape together. Because under normal conditions, if you think about it, in our body, the inheritance takes place all at once. It's not that one inheritance is taking place, then another, then another. It's like everything happens all at once, right? So now we're going to study the inheritance of two characters. So for this, a cross was made between a pure, that is homozygous, round yellow seeded plant. Round yellow, like round is the shape, yellow is the color. Both of them are dominant traits. Then wrinkled and green, both of them are recessive traits. So now you're going to take like capital R, capital R, capital Y, capital Y. This is the round and yellow. Cross it with small r, small y, which is wrinkled green. Now, from them, the gametes come out, capital R, capital Y, and small r, small y. These are the two gametes, isn't
Yes.
it? Now, when you cross them together, the F1 generation comes out to be capital R, capital Y, small y, isn't it? Because all of them will be heterozygotes. Now, so we had capital R, capital Y, crossed it with small r, small y, small y. From here, the gamete came out to be this, and from here, the gamete came out to be this. Now, when we crossed them together, the F1 generation came out to be this, right? This is the F1 generation. Now, in order to find out the F2 generation, we do selfing. And in order to do that, now since it is a dihybrid cross, you have to take out the gametes again, right? For the second generation. So the R with the Y, so capital R, capital Y, the R with the small y. So capital R, small y. That is done. Then this one. So small r, capital Y, and then lastly, small r, small y. So 1, 2, 3, and 4. These are the 4 sets of gametes that you have got. Clear?
Yes.
Now, in order to find out the F2 generation, you're going to write these gametes over here. If you want to, you can make the round, or it's like all right, you can just write the genotype also, that will be fine. So this is one set of gametes. You put a line like this. Okay, so here we say that this is the male set of gametes. Okay, below you're going to make the female sets of gametes that will be the exactly same thing. It will be capital R, capital Y, capital R, small y, then small r, capital Y, and lastly small r, small y. Understood? This thing that you have made, it is called as the Punnett square. Clear? This is like a checkers board to find out the genotype of the F2 generation. Clear? Up until now, the steps are clear to you?
Yes.
Okay. Now, how do you find out? You take this one, cross it with this one. Then you take this one, cross it with this one. Take this one, cross it with this, and then again cross it with this, like that. You have to cross each of the gametes with each of the other gametes. So for example, if you cross this with this, you're going to get capital R, capital Y, capital. Then second, you're going to get capital R, capital Y, small y. Then capital R, small r, capital R, capital. And lastly, capital R, small r, capital Y, small y. Understood? Now, same way you have to do for all of them. So now you only tell me this one.
Capital R, capital Y, small y.
Very good. Next.
Capital R, small y.
Very good. Next.
Capital R, capital Y, small y.
Very good. Next.
Capital R, small y, capital Y, sorry, small y.
Very good. Next.
Capital R, capital Y.
Very good.
Capital R, small r, capital Y, small y.
Excellent. Then
Small r, capital Y.
very good.
Small r, capital Y, small y. Capital R, capital Y, small y. Capital R, small y, small y. Small r, capital Y, small y. Small r, small y.
Okay, so these are the possible sets of the genotypes that you've got for the F2 generation. Now, once you find out the ratio, okay, so ultimately the phenotypic ratio of the dihybrid cross, it comes out to be 9:3:3:1. Okay?
Okay.
Which is like round yellow, then round wrinkle, round green, all the combinations that you get, that is the case. Clear?
Yes.
So now if you just look at these genotypes, can you tell me the phenotype of each one of them? The first one is round in yellow, isn't it?
Yes.
Then next?
Round yellow.
Next?
Round yellow.
Next?
Round yellow.
Next?
Round yellow.
Next?
What is the recipe for round? Wrinkled?
Wrinkled, huh?
Wrinkled and yellow.
Very good. Next?
Round yellow.
Next?
Wrinkled yellow.
Next?
Round yellow. Round
Next.
yellow. Round green.
Very good.
Round green.
Round
yellow. Wrinkled yellow. Round and green. Wrinkled and green.
So with this, you can deduce the phenotypic ratio. Any doubts up until now?
No, ma'am.
After that, there comes the law of independent assortment, which is in according to the dihybrid. So right now, we saw how two pairs of characters were getting inherited, right? Now we saw round, we saw yellow, and we also saw wrinkled and green. But there was no blending anywhere, isn't it? The round segregated independently. The yellow came out independently. So the law of independent assortment tells us that if two pairs of contrasting characters are present, each pair will segregate or assort independently from each other. There will be no blending, there will be no mixing. That round segregates from yellow. Same way, wrinkled segregates from green. It's not that one of the character is getting lost or it's missing out. Clear?
So isn't it same as law of segregation?
But law of segregation is mostly for one character. Here it is for two pairs. That is 4 characters all in one.
Okay, that is for one character and this is for 2 characters.
This is basically with like accordance to the dihybrid. That was with accordance to the monohybrid. Understood?
Okay.
After that, there comes multiple allelism. Now, multiple allelism is a phenomena wherein more than 2 alleles are present for one gene. For example, you saw that height was being represented by capital T and small, right? There were 2 alleles. But if more than 2 alleles are present, then we call it as multiple allelism. For example, the case of the human height. Okay, multiple allelism is like several genes, like for example, capital A, capital B, and capital C, capital C. This is the genotype of the darkest skin tone on the earth. Okay, so it's like more than 2 genes are governing the whole entire process. So that is what you call as multiple alleles. Clear?
Yes.
All right. Now, after that, we're going to see the chromosomal theory of inheritance. But for now, you note down till here, then we'll move. What is the last thing you've written?
Second. I completed with a question.
Question. Okay. Before that, before the question?
Before the question, monohybrid cross.
Okay. You write from test. All right, you can write.
Mom, could you show incomplete dominance? Yes, ma'am. Yes, ma'am. Co-dominance. Yes, ma'am. 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 Yes, ma'am.
Okay, any doubts up until now?
No, ma'am.
Uh, So now we're going to see the chromosomal theory of inheritance. Now see, at the time when Mendel was doing all of his work upon the hybridization and everything, so like even though he started the earliest, okay, like when no one was doing this thing, no one even had the idea of all of these. But Mendel's work was not at all recognized until the 1900, and he started his work in 1865. Why? It's because, first of all, there was lack of communication in his days, because of which his work was not widely published. Then he was the first person ever to use the mathematical knowledge, statistical knowledge in the field of biology, and that was actually unacceptable to the biologists of that time. And then he said all of these things, but it was like all theory. He could not give an experimental proof of the things that he stated, like whether it was law of dominance, assortment, anything of that sort. He could not give a proper proof. And he could also not tell that he knew that it was something that was being passed on, which right now we know that it was the genes, but he used to call them as factors, right? So no one knew what factors actually were. Again, chromosomes, the cell divisions like mitosis, meiosis, these things were not discovered during the time of Mendel. So then in 1900, three scientists were there, De Vries, Correns, and von Schermer. They independently came on to the same result that Mendel had already done. Okay, like they worked on inheritance and they came on to the same result. Also, by the time 1902, chromosomes were discovered. So it was much easier to study things because we know that chromosomes, they are the physical carriers of DNA, isn't it? DNA is, they are present on the chromosome only, and when the chromosomes get inherited, the DNA gets inherited. So Walter Sutton and Theodore Boveri, they said that the behavior of the chromosome and the behavior of the gene, they go side by side, they go parallelly. Like however the chromosomes behave, same way the genes would also behave. And this is how they use this whole entire thing to explain the Mendelian law. So according to Sutton and Boveri, the pairing and separation of a chromosome, if a chromosome pairs up or if the pair of the chromosome separates, together with them, the genes also separate or pair. Like however would be the motion or the movement of the chromosome, same exact thing you're going to find in the genes as well. Is this much clear?
Yes.
So now the knowledge of the chromosomal segregation along with the principles that had been laid down by the Mendel, the chromosomal theory of inheritance was laid out. And according to this theory, first of all, like how we know that when the DNA gets transmitted from one generation to another generation, the number, the structure, and the uniqueness doesn't change at all, isn't it? It's not that if the parent generation had this many chromosomes, the future generations would have lesser or more. That's not the case, right? However many chromosomes or DNA or genes are present in one generation, same will be present in the next generation. So likely chromosomes also, just like DNA, they have fixed number, fixed shape, fixed structure, and individuality throughout the organism's life. And it does not change, it does not get lost, nor does it get mixed up. Secondly, both chromosomes as well as genes, they occur in pairs. You know that like the normal body cells of the human beings or in general animals, of higher animals, it is diploid, isn't it? Diploid meaning two sets of chromosomes are there. It's the same way. The chromosomes as well as the genes, they always occur in the pairs in the body cells or the somatic cells. Then the two alleles of a gene pair, they are located on the homologous site on the homologous chromosome. See, homologous chromosomes are those chromosomes that are exactly like each other. They're similar in shape, similar in size. So the two alleles, they will be present on those homologous sites. And when the chromosomes would separate, the genes would separate during the gamete formation. And we know that the gametes, they get half the number of chromosomes, isn't it? So that necessarily tells us they will be getting half the number of genes as well. Much is clear? Any doubts?
No, ma'am.
Okay. Now this was the theory. chromosomal theory. And anything that is just theory is not, you can say, something that we can trust 100%. So experimental proof or experimental verification has to be given. And same way, the experimental verification of the chromosomal theory of inheritance. It was given by Thomas Hunt Morgan. And Morgan, for his experiment, he worked with the Drosophila melanogaster. These are tiny fruit flies. Okay, this is the scientific name of the fruit flies. Now the question comes along, out of all of the organism, out of all of the insects in the world, why did he choose Drosophila? Why not anything else? So then again comes the question, because you know that when you are testing any organism, you have to grow them also, isn't it? You have to add some character, grow them. So it's very easy to grow the Drosophila on the synthetic medium. Like you can provide media for that in the lab to grow the Drosophila. Like optimum condition, however, temperature, food and everything. Other than that, the Drosophila, it has a short lifespan. That is 2 weeks. So after 2 weeks, it will die. So you can have that much amount of work to do. That's it, and then you're going to take next reading. Also, after every mating session, the Drosophila produces hundreds of offspring. So it means that we have more this thing observations to take, and the male and the female flies are easily distinguishable. Just like how you can distinguish a male human and a male female human, same way the male and the female Drosophila can also be distinguished, and this property is called as sexual dimorphism, wherein you can actually differentiate between the male and the female of any organism. So here the males, they are generally smaller and the females are larger in size. And last reason to choose Drosophila is that they have only 4 pairs of chromosomes. That's it. Like that will leave someone. They just have 8 chromosomes to work with and that's all. Clear?
Yes, ma'am.
Any doubts?
No, ma'am.
All right. Now you can note it down. So this was chromosomal theory of inheritance that told us that the genes and the behavior of the chromosome, they go hand in hand. Clear?
Yes. Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am.
All right, Sanjita, let's just keep it till here only for today's class. And we're going to continue in the next class. Do you have any doubts up until now?
No.
Anything you want to ask?
No, ma'am.
Also, have you been receiving those homeworks that are sent after the class?
Yes, ma'am. The last time they sent monohybrid cross,
Hmm.
and in class
Hmm.
we like it was just written monohybrid cross, so I didn't know like what I mean how to
Like,
like.
did you open that PDF? The question might have been inside it. Like, when you open it,
No, ma'am. There's no question inside it. It was just written monohybrid cross.
like
I'll
in
sh-
your app,
yes. 1 second. This one.
and to open it,
It's like set pictures to upload.
there's no question given.
No, ma'am. It is just written monohybrid cross, nothing else.
There must have been some problem. Okay, I'll get it checked. Okay.
Okay, ma'am.
All right, dear. Okay, then I'll see you in the next.